All right, so recording here, and let's see. Stuff for today. I will remind you that regarding the Blackboard quizzes for the rest of the semester, I'll just be sending out email notifications when they are posted, including the due date, and I'll basically be giving you at least 4 or 5 days after the email is sent. And I intend to finish one or two of those tonight. So please be keeping your eyes open for those emails, and there's not that many more of them to go. Let's see. Additionally, we're going to wrap up the ERP unit today, and we've learned there's been a lot of stuff that we've talked about. These slides ended up being like 50 slides long. We've been doing it for basically 2 weeks, and we've gone through a whole bunch of different event-related potentials. And I don't know if some of you guys probably have taken cognitive neuroscience, but we cover in this course way more event-related potentials than are done in the cognitive neuroscience course here. So this is a pretty good survey of the big event-related potentials that occur in the current cognitive neuroscience literature. And we do have, I have an in-class discussion quiz following the lecture today that I'm going to make due tomorrow at 11:59. 2 questions, but they're a little bit more involved questions than usual. So that's the plan. I will encourage you guys, you're still working on group project 2. I know a lot of the groups now are finishing up coding, and I've looked at some of the codes and they look good. And I'm pretty pleased. It's the first time Toolkit has been used in this course, and it seems like it's working. Your guys' scripts, your code looks good, and the experiments themselves are cool. If there are groups that have not met with me and would like to, regarding the scripting of your code in Toolkit, I'll remind you I'm definitely happy to meet with you. And I've been giving extra credit. I gave 3 points to people who have met with me previously, up to 3 points once you have met with me and your code is good. And I'm going to continue that offer. I'll still give extra credit. If you haven't met with me yet, I'll still give extra credit. At this point, I probably won't be giving you the full 3 points if you're just now notifying me. But I still want people to meet with me to talk about your code for Toolkit. And if you haven't emailed me yet, definitely feel free to send an email, like an appointment, and I'll still give you like up to 2 points of extra credit for meeting with me to discuss your code for group project 2. And as usual, I'm general to answer any questions about group project 2 that you might have. So yeah, we'll start talking about, let's see, we've gone through a lot of these tightly time-locked event-related potentials. We ended up last time, we've basically finished talking about the P300. I'll just briefly say the last thing we were talking about that. And then we should be able to get through, I spent a lot of time talking about these at the beginning. We should actually be able to go through these the plan is to get through all of the remaining event-related potentials today. So we won't be talking nearly as long about these per event-related potential as we did these at the beginning. So starting with the N400, well, actually, we'll start with the P300, which we already talked about, just reviewing it. We talked about it with like 3 different experiments, the P300. And this is one of the ones that we talked about. I added a bullet point here just so, because there wasn't when we talked about this last time, there wasn't really a conclusion bullet point on the slide. So I have added that over the weekend, and I've, of course, have updated, I've overwritten the previous file. So there's an updated set of slides for this unit. And those should be the last updates for these slides. I've been updating them each time. This should be the final update for this unit. So this is the study we talked about last time. They measured the P300 during the Stroop task, which is something we know a little bit about. They added a condition that we didn't have for our Stroop task that we've done in this class. We just had the congruent, or what they called compatible, and incongruent, what they called incompatible conditions. They added this unrelated condition. And then, as I mentioned here, there's something weird about that, because usually the unrelated condition, the reaction times are usually not that slow for the unrelated condition. So there's something weird going on here. What unrelated or neutral condition, usually the reaction times are quicker there than they are for the incompatible. It's very rare that this would be I've never seen a study other than this one where the unrelated condition has the slowest reaction time. So I think it's possible that they might have like reversed those or something, or made an error when they were compiling the data. Don't know, but something's a little bit fishy. But the reason I do like this is because these results in the bullet point here kind of support this general idea that we've been talking about with the P300 event-related potential. One of the things that it's thought to reflect is decision-making processes. You have some stimulus and you have to make a decision about that stimulus. The P300, properties of the P300 appear to reflect some of those mental processes that are occurring while a person is making some judgment about a stimulus. And that kind of idea is supported here in this P300 study of the Stroop task, because right here, the latencies tend to be longer for the incongruent condition. So this is P300 latency, that positivity. It's called the 300, implying that the latency is usually 300 milliseconds. But as I mentioned, the latency of the P300 actually ranges between 250 and sometimes up to 900 milliseconds after a stimulus. Here, though, the dependent variable is the latency in milliseconds. And you can see that the latency of their congruent condition is definitely small. So the P300 latency, that means that it peaks earlier on. It peaks sooner than the P300 when it peaks in the incompatible condition. What's nice about this is this appears to kind of reflect the decision-making process that occurs and which is evident in reaction times in the Stroop task. And the reaction times of Stroop task, they tend to be bigger for the incongruent condition, and they tend to be smaller for the congruent condition. And you see a similar thing here for the P300 latency. And so that's kind of nice when you can see this correspondence between what behavioral measures like reaction time show and event-related potential measures, in this case, the P300 show. So this is where we left off last time. The N400, it is a negativity with a latency of 400 milliseconds. And it occurs, the negativity is larger in response to a word which is a violation of your semantic expectations. So there's two different paradigms in which this is often used. One, when there's just a list of words that are presented. And then at the end of the list, there'll be a word which either fits semantically with the previous words in the list or does not fit semantically with the words in the list. And I think I have some examples here. Yeah. So for instance, the first word would be cat. And then the researchers would measure the event-related potential response to the word dog. That would be related. That is, these two words are thought to be semantically related because they both fit in the category of domestic pets. And that would be the blue line right here. So the name of the word dog would be presented at the 0 time point. And then about 400 milliseconds later, that's the response of interest in the electrodes. And then the response to the word dog is more on the positive side of things relative to this red line. This red line is if you present the word son and then you present the word dog, these two words are not that semantically related. They're two different types of things. They don't really fit in the same category. They're certainly far apart. Son and dog are farther apart from each other than cat and dog. I think that's pretty clear. And if you look at the response to the word dog, when it's preceded by the word son, you can see that there's this more negative pointing wave right here. And so at around 400 milliseconds after the word dog, when the preceding context is unrelated to the word dog, that is the word son, you get a relatively bigger negativity than when the word dog is preceded by a related context, that is the word cat. So the N400 is right here. It is a negativity about 400 milliseconds after a word. The amplitude of the N400 is bigger following a word which is semantically unrelated to the previous context. So this is an example of context where the context is just a word, or sometimes they'll have a longer list of words. In this particular study, it's literally the context is just one word that precedes the target word. That's a pretty common way of doing it. The other way of doing it is shown in this video, which I'll show you right here. So before the pandemic and all this happened, there was actually, we got some new equipment for the lab, which we were going to do some cool labs. And unfortunately, we weren't able to do it because we have to, social distance and stuff. And so we couldn't use the lab space. But I'm going to show you what the data collection for this lab would have looked like. So I used myself as a participant. And it shows us, it shows the experiment that I created that measures event-related potential responses in the N400 paradigm, but using sentences. And so I'll show the video, and Yeah, I'll just go ahead and show the video right now. And I'll kind of explain things as they occur in the video. So there's a couple of things to take a look at here. So one, I don't know how good the screen clarity will be as I'm sharing this and showing you the video live. So I do encourage you, the link should work for you to click on the link and then watch this on your own time. But there's a couple things that I want, and I will show it to you now anyway. There's a couple things I want to point out. Up here, these red and blue traces, this is the raw EEG, but it's from two different electrodes. Here it's called electrode 1 and electrode 2. So this is just the electrical activity at two different points on the scalp, excuse me. And those are being recorded while the participant is reading sentences right here. So I'm using these are actually two different computers. We use one computer to measure the electrical activity, and then we use another computer to present the stimuli, and the participant is looking at the screen right here. And so the sentences that the participant reads are right here. And once I press play, you'll see the words starting to flash. The participants are reading those sentences. Some of the sentences, the target word is semantically related to the previous sentential context. In some sentences, the target word is not related to the previous words. And so I'll show you that. One of the things that I want to draw special attention to is there's going to be those little markers that appear down here. And those markers correspond to the exact point in time in which the target word is presented. And then, yeah, so I'll show you, I'll just go ahead and show you, and I'll kind of talk as it's going along. So I'll press play. Please be kind of looking here. You kind of need to be monitoring two things. So I'll probably press pause at one point, and I'll be reading the sentences as they appear down here. Okay, it's starting now. Press spacebar to continue to next trial. She grew a plant in the pot. Okay, and this is a marker right here. This is the exact time point when the word pot was presented. And this sentence was intended to be one in which this is the target word was pot, in which the target word was considered to be related to the preceding context. And so what we would do is you would take this time point in the event relay in the electrical data, and you would take all of the amplitudes right here. And this is like trial 1. You would take this, and this would be like the first trial of the related condition. And then there'll be other trials, which are other sentences where the target word was related. At the end of the experiment, you add up all of the EEG data, which occurs at these exact time points relative to these markers that will occur when the target word occurs. You basically average those together, and that is the event-related potential amplitude for this, the related condition, because, you know, pot is something you would definitely plant something in. That definitely makes sense. So then we'll go to the next sentence. Press spacebar to continue to next trial. She grew a plant in the fridge. So now this is the marker, but you can see it's a different color, and it's also a different channel. This blue marker right here, this represents the exact time when the target word fridge occurred. So that would be the unrelated condition, because the word fridge, that does not fit. You don't grow plants in fridges. That's pretty unusual as far as I understand. And so this would be considered an unrelated. This would be the condition, by the way, where you would expect the N400 amplitude to be bigger. You'd expect the negativity to be bigger here. The way we do it is you would take this exact point in time. You would take the electrical activity in a certain time window relative to the onset of that word, and this would be the first trial of the unrelated condition. Then you'd have other trials for the unrelated condition, and you would add up the amplitudes at the same time window relative to all of those different target words. You'd add them all together, average them, and that would give you the wave. That would give you the amplitude, the event-related potential response for the unrelated condition. So I'm going through that a little bit quick. I do suggest you take a look at that on your own, because I think this conceptually, if you're struggling understanding what event-related potentials are measuring, this right here, like this is probably the most direct, concrete thing that you could use to kind of help understand how exactly event-related potential paradigms work, because this is literally one of the ones which is used very commonly in the literature for the N400. Okay, so that's moving on. P600. This is kind of similar to the N400, except instead of it being violations of meaning, violations of semantics, which is what the N400 is, the P600 is violations of syntax, or maybe more commonly known as grammar violations. So this is one presented here. It's shown in, it was originally presented in German, so the translation is shown in English down here. So there's like two conditions, the grammatically correct and the grammatically incorrect condition. The grammatically correct one would be the spoiled child throws the toy on the floor. That's grammatically correct. The incorrect, grammatically incorrect condition would be the spoiled child throw the toys on the floor. So this is like, there's many aspects of grammar, but this would be considered subject-verb agreement. Here, the verb either does or does not agree with the subject of the sentence. This is the subject. These are the verbs right here. One of the verbs agrees, the other one doesn't with the subject. And then the researchers compare the brain's response to these words. These are the target words. That's why they have stars next to them. These are the target words right here. And the researchers measure the response to these words. So in this, these are sentential context. So they, like, you're listening to a whole sentence, then the target word appears, and then that's the word that the researchers are interested in the amplitudes of the different conditions. So in this particular trial, this is one long trial, at 1200 milliseconds after the beginning of the trial, that's when the target word is presented, either throws or throw. And then about 600 milliseconds later, that's like the lag, that's the latency. 600 milliseconds after you hear the verb, that's when your brain processes or is representing the difference between the verb when it's correct grammar and the verb when it is incorrect grammar. The P600 is the positivity that occurs 600 milliseconds after this that is bigger in response to the grammar violation, for the word that violates your expectation regarding the grammatical structure of the sentence. And so I know some students have struggled with this graph before, so I'll point out positive is down, negative is up. The P600 is this right here. And the reason this is, it says like 1800, and that might be confusing. The reason this is the P600 is because 1800 is exactly 600 milliseconds later than 1200. So these are when those target words are presented. 600 milliseconds later, that's when the brain is representing those verbs. And you can see that grammatically incorrect, that's the blue, has a relatively bigger positivity than the grammatically correct, which is the red. Any questions or comments about N400, P600? All right. Okay, so motor event related potentials. These are cool. I chose not to have you guys read an article on these, but they're pretty straightforward, I'll say. And so I'll just kind of show you the data from this one experiment, Tarka et al., 1994. So motor event related potentials, these are basically, there's kind of like two things which are studied with respect to these. One, literally what the brain is doing when you're making a movement. That's a motor event related potential. But then another part of these things which is oftentimes studied is what your brain is doing in preparation of a movement. So you're about to move. What is the brain doing when you're preparing to move? And what does it actually do when you actually move? So in this particular study, it was self-paced, that is voluntary movements. Basically, the participants make, they just kind of at random points in time decide that they're going to make a movement. And then the research measure what's happening in the brain around the time point where they're making this movement. And there's a couple of findings. Preparation to move. So this is like right before you move, but you know you're about to move. So you're just sitting there and you're about to move. And then your brain starts to change. Like the brain's not really doing anything, at least in the specific electrodes. But then you're about to move. And then the brain starts before you actually move, you can see that there are changes which are occurring in the brain. They kind of predict this movement, which is going to happen in the very near future. And those changes that occur right before you move, but when you are preparing to move, are typically greatest over midline electrodes. So meaning like right in the middle, top part of the brain. There's a lot of activity that occurs in preparation of a movement. Though I will also point out, if you're moving, like in the graph I'm about to show you, it's a movement of your left hand. The participant is told to move their left hand on this trial. When you're preparing to move the left hand, there's a lot of activity midline. So it's like the middle of your brain knows before that you're preparing to move your left hand. So the most activity is midline. However, there's also a tiny bit of activation and preparation contralateral to the movement of this left hand. So contralateral to the left hand would be the right hemisphere. So you'll see, I'll show you the data in a second, a lot of preparatory activity in the midline, but there's also a little bit of preparatory activity in the contralateral hemisphere. And so these are generally suggested, this midline effect where you see a lot of amplitude and preparation of a movement, generally thought to rely on this part of the brain called the supplementary motor area, which is shown in purple right here. And as you can see, it's in the middle of the brain. There's one in the left hemisphere, one in the right, but they kind of straddle the longitudinal fissure, one on the left, one on the right. And that's thought to be where kind of these preparation type responses, when you know you're going to make a motor movement in the near future, but before you actually make that movement, the supplementary motor area appears to be part of the brain, which is helping you prepare, maybe organize or plan the specific things you're going to do with your motor system. And then the movements themselves. So once again, all the stuff I was just talking about, that was like before you move, the preparation to move. But the movements themselves, the activity was generally greater in the contralateral hemisphere. So I'll show you both of this stuff right here. This is abstract, actually. It's a little bit wordy, but if you kind of ignore the overly wordy stuff, like there's like a wordy sentence, and then they'll have a sentence which is like accessible and makes sense. Then they'll have another wordy sentence, right? What the heck are they talking about? Then there'll be another sentence which makes sense. So if you can kind of sort out the stuff which is meaningless and the meaningful stuff, this abstract actually can be kind of helpful to you, which is why I've included it. Here are the data for their experiments, though. And so this is a little helpful thing to look at. For one thing, I'll point out these are left-hand responses, so they're moving their left hands. And this 0 time point, which is where these dash marks in, each of these things is an electrode. This dash point, that is the 0 time point. That is when the movement occurred. So they're calculating event-related potential. They look at when a movement occurred, and then they're using a time window to measure the electrical activity for time preceding that movement and for time following that movement. So they're wanting to know, what is the brain doing before you actually move? What is the brain doing slightly after you move? And of course, what is the brain doing when you're actually performing the movement? And so when you're looking at the activity of the brain when you're preparing to move, basically what the question is what does the brain look like those few milliseconds before you actually have moved? And so to do that, what you do is you start here at the dash point, and then you look backwards in time. You look into like the negative points of time, which are represented here. And as you can see, if 0, like this horizontal line here, that basically means no electrical activity, or no difference in electrical activity. That's like the baseline activity. But as you can see, these are the midline electrodes right here. And you can see that this is non-zero right here. This wave is above the 0 line, and this is before the movement actually occurs. So in particular for the central midline electrode, CZ, which is the dead center, it's like the equator and the equator and the prime meridian basically of your brain, like right center part up here, there's a lot of activity that occurs before you actually make the movement. So this is thought to originate from that supplementary motor area, because it's right there underneath the scalp, right there, right in the center part of the brain. And so that's the preparation movement. But please also note that if you look in the right hemisphere, there's also, so these, now we're getting in the right hemisphere. These electrodes over here are lateralized to the right, on the right side of the scalp. And you can see that even before the movement in the right hemisphere, there is some preparation. So the preparation seems to definitely involve this center part of the brain, but it also involves the part of the brain which contralateral to the movement itself. An important comparison here is, what is the left hemisphere doing? And if you look, the left hemisphere really isn't doing much of anything, not during the preparation, and it does maybe a little bit in response to the movement itself, but not much. And it's actually possible that this little bump we're seeing here is actually electrical activity from the right side of the scalp, which just makes its way over to the electrodes on the left side of the scalp. And so the activity in the left hemisphere is generally less, both in preparation of the movement and performing of the movement itself. The preparing to move definitely shows some midline activity. And it also shows, and the actual movement itself also shows some midline activity as well. But the strongest movement, the strongest activity occurs during the movement, when you actually perform the movement, and it's contralateral to the part of the body which is performing the movement. I guess the main takeaway here is that the preparation to move seems to involve these midline structures a little bit, possibly the supplementary motor area, but the actual performance of a movement appears to rely most strongly on contralateral parts of the brain. I guess I should show you this primary motor cortex. You probably have learned about this in a different psychology class. This is the kind of famous homunculus. It's the part of the brain that when you stimulate it electrically, you can cause different parts of the body to make little jerking movements, like I just did. If someone stimulated my left motor cortex, they could make my right side of my body kind of flail a little bit. You can make me blink or move my arm or move my leg or move my foot, depending on which part of this primary motor cortex is stimulated. The primary motor cortex is thought to control the opposite side of the body, the contralateral side of the body. Whereas, once again, the supplementary motor area, which is middle, it's central, is thought to be most important for kind of planning to move. So those are motor event-related potentials. So here's the error-related negativity. You read an article about this one. So this is an event-related potential which occurs when after you make some type of an error. So it's kind of like an error detection system that your brain has, that right after you make an error. So I think the best example, and it is a commonly studied type of an example, not the example I provided, but like you're typing on a computer and you kind of know what you're trying to type, but then you kind of like know when you make an error soon after you make it, even if you don't necessarily see the error on the screen. You kind of like, you meant to type a letter, and you're supposed to do a certain type of movement, and you know that you didn't do the right movement. Like, you're trying to type an S, and you know that you accidentally typed an A. That's like a common thing that I'll do. I'll try and type an S, but I type an A because they're right next to each other. And then, there's other type of errors you can make, of course, in a keyboard. The error-related negativity is the idea that you kind of know that your motor system did the wrong thing. Your motor system was supposed to do one thing, but it did something else. So in this study, participants were trying to respond. I think they had left-hand response, right-hand response, and they were squeezing these things depending on the different type of letters that would appear on the screen. And if one letter appeared on the screen, you're supposed to squeeze your left hand. If a different letter appeared on the screen, you're supposed to squeeze your right hand, and you're supposed to do it as quick as possible. And sometimes people made errors. Sometimes there would be something on the screen and you were supposed to squeeze your left hand to give an answer, but you'd accidentally squeeze the right hand. And kind of, you're trying to do it really quickly. And so sometimes you do it too quick and you make the mistake, but your brain kind of knows, oh, that was the wrong thing. I wasn't supposed to have done that. And you can tell that the brain knows that you did the wrong thing by measuring the amplitudes of the brain in these two different conditions, those conditions in which you did the correct thing compared to those conditions when you do the incorrect thing. And that's shown here. The incorrect response is this dotted line, and the correct response is here. In both these situations, you're squeezing the hand muscles, but in the incorrect, you're squeezing the wrong hand muscle, and the correct, you're squeezing the correct hand muscle. And the error-related negativity is the idea that there's a negativity that occurs usually in a pretty small time window. Here in this study is like 100, 120 milliseconds after the behavior. So EMG onset, this is a way of measuring muscle activity. And we'll actually be talking about that for this last week of class. We'll talk a little bit about EMG. It's another physiological measure. And it measures basically when the electrical activity in your muscles fire action potentials. And basically when there's a peak, they're measuring from your arm muscles, whichever muscles are involved with doing that. And then when they find that there's a peak, which is above some threshold, they know that your motor system is doing something, and then they'll measure the electrical activity coming from your brain in the time window right after that. And when those muscle movements reflect an incorrect response, there tends to be a negativity, much higher amplitude negativity relative to those trials in which there's a correct response, which is a relative positivity. So the difference in amplitude between these two conditions, that is the error-related negativity. All right, so those are all of the tightly time-locked ERPs. So I told you we'd be going, the first few, we took a long time kind of fleshing them out. We've gone through a bunch of those tightly time-locked event-related potentials. And now we're starting to get into some of the less tightly time-locked event-related potentials. So these are those where they're kind of now looking, generally they're looking at the electrical activity as it spans over a relatively bigger point in time. So for a lot of these event-related potentials, the window is usually like a very small window of time when the researchers are measuring the amplitudes or the amplitude difference of interest in those tightly time-locked examples. Those time windows are typically pretty small. For the ones we're going to be talking about now, though, the window of time that the electrical activity measured is generally a little bit longer. See, these are kind of the exceptions to the rule. These are important event-related potentials. They're just less tightly time-locked. Usually the window of time when things are measured is just bigger. Okay, so the contingent, so again, there's two of them talked about here, and I'll go into details of each one. These are typically categorized as steady potential shifts. I don't actually care about you knowing that, but that is a terminology which is oftentimes, in textbooks at least, describes these two different event-related potentials right here: contingent negative variation, or CNV, and then the readiness potential. These are very similar, and we'll talk about how they're similar, and we'll talk about how they're different. They're both negativities that occur, but the CNV, like in the CNV, it occurs after some warning signal, but before a probe. So the warning signal basically says, in the near future, you're going to respond to a probe or a test stimulus. The warning signal says, get ready. And then the probe is like, okay, this is the thing that you're getting ready for. This is the thing to which you're supposed to respond. And the contingent negative variation is the idea that once that warning signal occurs, there's this general increase in negativity that occurs until the probe occurs, and then the negativity goes away. So it's like a buildup that occurs in anticipation of something. And that anticipation of something occurs because of this warning signal, which you are provided. So actually, let's go talk about that. Then we'll come back and I'll talk a little bit more about readiness potential. So the CNV, contingent negative variation, it is a negativity. Some common terminology are this S1 and S2. The S1 basically just means warning signal. S2 is sometimes called the imperative stimulus. I think in the previous slide, I called it the probe stimulus. Sometimes it's called the test stimulus. Usually, though, I'm actually going to present a study where for half the trials, it did not require a response. So the S2, the target, the probe, or the imperative stimulus, S2, it's got a bunch of different names. Usually the participant has to respond to that. Sometimes, though, the participant will just look at it and not do anything at all. So as I already mentioned, that CNV, it's a negativity that builds between those two stimuli, the warning signal and then the probe stimulus. And it is strongest at CZ. So once again, those midline electrodes. So there's kind of varying theories about this, but I actually believe that this CNV signal is probably coming from the supplementary motor area, that midline area, which is involved with motor preparation. That's what I think is going on here, although it's debated. But the signal is strongest. You get the biggest CNV, contingent negative variation, and CZ, which is that very central electrode right in the center of the scalp. Though you do find the response in different electrodes. It's just biggest right in that middle part of the scalp. And here is an example. So I have this is the abstract that goes along with this. Once again, there's some somewhat helpful information in this abstract, though I'm going to try and describe it in even simpler terms right here. Yeah, I didn't actually mention this here, but the C and V is one event-related potential, which also can be kind of like a biomarker for SZ, which stands for schizophrenia. So it's another one of these event-related potentials, which is different between healthy controls, HCs, and individuals with schizophrenia. And you can see that right here. So this study is a little bit complicated, so I'll have to kind of explain things a little bit. First off, I'll point out that negativity is down, positivity is up. And so this Q on, so they're using slightly different terminology, which is oftentimes taught in textbooks, which does happen. Q on, basically, this is the warning signal, S1. This basically means, okay, something's about to happen. And then that something that would happen, the Q would stay on for about 3,000 milliseconds. And during that time, that's basically the warning signal. And then the stimulus that you are supposed to respond to in this case is when that Q turns off. And then when that happens, the participant is supposed to perform a motor response. They're supposed to press a button on a keyboard. So this is a little bit weird because oftentimes the warning signal will be one thing, and then the target, S2, will be a different thing that occurs. In this case, the S1, or the warning signal, is just a cue that turns on and it stays on the screen for a while. And then the S2, or the target, is when that cue turns off. So when the cue turns off, that means you're supposed to respond by pressing a button. The important stuff is what happens in between these two things. Right here, this is when the cue turns on. And then, as I mentioned, the C and V is that gradually building negativity that occurs in anticipation of performing some type of action, or in anticipation of seeing some type of stimulus. And if you look here at the healthy controls, that warning signal occurs, it turns on, and you can see there's some, a response to the stimulus itself, but then there's kind of the steady potential shift that occurs where the electrical activity in your scalp is just going negative, 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 just keep getting more and more negative until the actual S2, in this case, the Q turns off. That is the S2 in this study, until that occurs, in which case things return to baseline. The green line is the healthy controls. The pink line here is the individuals with schizophrenia. And as you can see, the individuals with schizophrenia, they do show that C and V. They do show the gradually building negativity, but the amplitude is clearly different. Both controls and schizophrenics have around the same amplitude at baseline. But then during that waiting period, the preparation period, the schizophrenic's brain gets less negative, whereas the control brain gets relatively more negative. But then when the S2 occurs, in this case, when the Q turns off, the brains get back to similar. The schizophrenics and the healthy controls, their responses are the same here. So this is probably the most, this part of the experiment's pretty straightforward. The thing which makes this one a little bit confusing is that the Q turns off. This is the S2 for the first part of the experiment. That is, this is the probe for this first part of the experiment. But then it also serves as the S1 for this next part of the experiment. So as soon as the Q turns off, you're supposed to hit a button on your keyboard, the space bar. But then that immediately starts the next trial. So this Q turning off is then helping you prepare, or it's the warning signal for this picture, which is going to occur, like 3 to 4 seconds later. And in this picture, though, it does not require any type of motor response. So the Q turns off. This is the warning signal then for this picture of the picture that occurs later, though this picture does not require any type of response on your keyboard. You just look at it and you're like, okay. And if you notice, you still get a contingent negative variation here, but you can see that the amplitude of the C and V is way weaker in this situation than it is here. You can see, by the way, the healthy controls still have a stronger C and V in this situation than the schizophrenics. This green line is below the pink line, meaning the healthy controls have stronger C and V. But if you compare the amplitude of the C and V overall in this condition to over here, the C and Vs are much bigger. Over here, they're not as big. And one interpretation of this, which I believe, is that the C and V is particularly strong, or the main purpose of the C and V is to prepare your brain to perform motor movement. Here, this is when you are performing a motor movement. When the cue turned off, that's when the participants had to press a keyboard. In anticipation of having to press the keyboard when that target signal occurred, that is when the cue turned off, that's when the C and V amplitudes overall were biggest, particularly for the healthy controls, because there was a motor movement. Over here, though, the target stimulus, the probe, the S2 over here, did not require any type of motor response. You just passively view it. You see a C and V, but it's not very strong. So there's a couple things, couple punchlines here. In general, the amplitude of the C and V is stronger when you have to, in response to the S2, the probe, the target. It's stronger when you have to produce a motor response to that S2 stimulus, which is over here. The other takeaway is that the CNV is a pretty effective biomarker for schizophrenia, such that the amplitude of the CNV tends to be smaller in individuals that have schizophrenia compared to control. So that's the CNV. And I mentioned it's similar to the readiness potential. The big difference, both of these are negativities that occur in anticipation of some type of, usually it's a movement. The difference is, though, is that in the readiness potential, the movement is voluntary. It is self-paced. You decide when you're going to move. In the contingent negative variation that I just showed you, that's not the case. There'll be a warning signal which says, soon we're going to make you move. And then that S2 would occur, that target, that probe, in the contingent negative variation, that occurs, and that's saying, okay, do your thing now. So the contingent negative variation, the participant is kind of responding to stimuli out there in the world or on the computer screen. And the readiness potential, though, on the other hand, it's completely voluntary. Oftentimes, the experimenter's instructions in the readiness potential will be, make a movement with your left hand when you feel like it. That's a common instruction. So it's when you feel like it, it's self-paced. You decide when you're going to move, or voluntary. And the readiness potential is the fact that there is, before you make that voluntary movement, there is anticipatory things which are occurring in the brain for about 500 to maybe 1,000 milliseconds before you make that movement. Your brain is kind of getting you ready to make that movement slightly before you make it. So you can look at the brain and you can actually tell, like 500 to 1,000 milliseconds before a person makes a movement, that they're probably going to be making a movement in the very near future. And I'll show you that as well. Yeah, here it is right here. So yeah, it's also called the Bereitschaftspotential. Sounds German, I believe. I don't remember the person in the name. We just get about German researchers presumably discovered this. So it's also called the BP, the resting potential, or the Bereitschaftspotential, BP. Here I kind of define it again. And it does have a similar scalp topography as the C and V, meaning the amplitudes tend to be largest at these central electrodes. So once again, this readiness potential probably uses that medial structure, the supplementary motor area, which is kind of in the middle top part of the frontal lobe over here. At least that's what I think. SMA is probably a main source for the readiness potential and the C and V. And here, this is the graph I care about you knowing. These other graphs I didn't cut out. But you should ignore all these. The only one I care about you knowing for this class is graph A. And so in this graph, negative is up, positive is down. At 0 time point, this is when the participant does that voluntary movement. They move their hand, or they usually, that's usually what it is, move your hand. And then, so 0 time point, that's when the participant actually performs the movement. But you can see that up to that time point, for about a second, actually it looks like it's maybe even like a second and a half here, you can see that gradually building negativity that occurs up to the point in which that movement occurs, in which case things kind of, at least in these medial electrodes here, things go back down to kind of the baseline level. So it seems to be like an anticipatory brain response. Your brain kind of knows that you're going to move slightly before you do the movement itself. And so I mentioned, way back here, in this, and I'll go into more details about this. This is probably the most famous readiness potential study by Libet. I believe I'm pronouncing that correctly. And it's a study which the results of the study are oftentimes suggesting something which does not sit well with a lot of people, which is we don't actually have free will. And I'll explain why. I'll explain that, how they got to that interpretation here. Kind of the overall punchline of the findings is that the readiness potential precedes the time point in which participants report they got the conscious urge to move. So the participants say, okay, that is the exact time point where I decided I wanted to move. But then if you look at the brain activity, the brain actually starts showing evidence of the readiness potential before the participant even says, that is what I felt that is when I got the exact urge to move. And so I want to actually go into a little bit of the detail about how do you get that? How do you study this type of thing? How do you know when the participant how do you know the exact point in time when the participant has the urge to move, when they decided to move? Let's see, hopefully we have that here. Yes, here it is right here. So here is the famous Libet study on free will. So once again, participants were making self-paced hand movements. Move your hand whenever you feel like it. The readiness potential was recorded. And while they were doing that, participants were actually, so this was done a while ago, and the participants were actually looking at the second hand on a clock that was moving. And then, participants would make these self-paced hand movements. They would just move whenever they felt like it. And they would note the time by looking at that second hand on the clock when they got the urge to move their hand. And as I kind of already mentioned, the rise of the readiness potential, so that negativity started to occur after the time point when participants felt they had the urge to move. Wait a second. The readiness potential occurred, yes, after the time. So participants said that I'm going to move here. I saw this as a typo. The readiness potential occurred before the time point that participants stated they felt the urge to move. So that is a definite typo. The readiness potential occurred before the time point, and I said this earlier correctly in the previous slide. The readiness potential occurred earlier than the time point which participants felt that they were going to move. And so it'll be most easy if I show you with respect to this graph right here. So here's the readiness potential occurring right there. That's the building of negativity. While, this is time on the x-axis right here. And while participants were just randomly making movements, they were watching the second hand moving on the clock. And then at some point they would note the exact time point on that second hand when they're like, okay, I'm going to move now. And then they would remember that number, and then they would report it to the researcher. So they'd say, when I decided to move my hand on that trial, the second hand was at this exact time point. And then they would look, what was that time point that they said relative to the actual electrical activity of the brain? And what they found was that that time point would oftentimes be like right here. That's when the participant would say, I decided right here that I was going to move my finger. But if you actually look at the brain's response, the brain started showing evidence that you were going to move earlier than that. So you said, this is when I decided at this time point, this is when I was going to move. But the brain already kind of knew you were going to move before you actually did it. So let's go back to this. The rise of the readiness potential occurred before the time point that the participants stated they felt the urge to move. And so what this suggests is that your brain kind of knows that you are going to do something before you do. You said, I decided to move right here at that point in time, but your brain is actually saying, nah, you actually decided you were going to move back here. And so the popular interpretation of this is that the brain knows what we're going to do before we do it. And, skeptics or people that say we don't have free will at all will say that free will is an illusion that actually your brain kind of makes these decisions for you, and you feel like you're in control of your own movements, but actually the brain is making all of these decisions for you. And then a split second later, that's when you feel like you're making decisions. That's a common or a popular interpretation of this study. So yeah, the rise of readiness potential occurred before. I had a typo. It definitely occurs before the time point when participants felt they had the urge to move. And then this is the last one, the last event-related potential. It's another one of these not tightly time-locked ERPs. The steady state evoked potential. It's kind of the exception to the rule of all of these ERPs. I mentioned at the beginning of this unit that one of the defining features of event-related potentials is they don't involve frequency analysis. And for the most part, that is true. But steady state evoked potentials are a pretty big exception to that. These things are definitely use frequency analysis. Remember, frequency analysis is basically taking the raw electrical activity and decomposing it into its different sinusoidal components. And so evoked here, which is in the name, that means that this is the brain's response to a stimulus out there in the world. But that it's important because contrasting that with an invoked response, evoked versus invoked response, which is based on a mental process. So evoked is your brain's response to the to a stimulus which is out there in the world. And it's those aspects of the response that don't require any type of mental processing. It's just kind of encoding the stimulus, its presence itself, without actually any type of mental computations of that stimulus. That's what evoked is, compared to invoked, which is a mental process when you're somehow making some type of a computation or thinking or making some type of a decision. So an example of an invoked oscillation or an invoked wave would be when you're retrieving something from long-term memory. One of the neural correlates of that's been studied is the fact that there usually tends to be an increase in theta band amplitude. That's a mental process. Retrieving something from memory is a mental thing. Steady state evoked potentials, these aren't about mental things at all. This is just the response of your brain to some property of the stimulus without any type of mental computation or without any memory retrieval or anything having to do with, thinking at all. Typically, in fact, the only time I've ever seen these is steady state evoked potentials respond to the timing properties of a stimulus. So in particular, they'll present a stimulus which is flickering, kind of like a blinker, but it can flicker slowly. That'd be like, 1 or 2 hertz, or it can flicker fast. I can't do 10 hertz because that would be 10 things per second, but it flickers basically a certain amount of times per second. And that's what hertz is. The more times it flickers per second, the higher the hertz, the higher the frequency. The steady state evoked potential is the fact that you can actually look at the electrical activity of the brain, and then you perform a frequency analysis. And what you tend to see is that if the stimulus out there in the world is flickering at 5 hertz, then when you perform the Fourier analysis, when you perform the frequency analysis, you're going to see a peak in amplitude at 5 hertz. If the stimulus is flickering at 10 hertz, 10 flickers per second, and then you do the Fourier analysis, you do the frequency analysis, you're going to see that there will be a peak in amplitude that occurs at 10 hertz. And I have some graphs that will help kind of explain, that kind of demonstrates this. Yeah. Right here. So this is like what the EEG activity actually looks at, like the actual waves. So this is like a stimulus flickering 10 times per second. And then if you actually look at the electrical activity of the brain, it's kind of got this oscillation or this wave to it. And the wave, if you actually do the Fourier analysis, this is a Fourier analysis down here. The x-axis is the different frequency bands. And then the y-axis looks like it's power here because it's squared. Power, which is like amplitude, is amplitude squared. And you can see that there's a peak amplitude, a peak power that occurs at 10 hertz. That's what we're seeing right here. And another example, though, here's a stimulus which is flickering at 20 hertz, so relatively faster. This is what the EEG looks like. If you perform the Fourier analysis, if you perform the frequency analysis, you will see that there is the biggest peak occurs at 20 hertz. So this is basically the 20 hertz oscillation. This is a 10 hertz oscillation. But it's important to notice that there are small peaks that occur usually at integer multiples of the original evoking frequency. So 10 hertz, the biggest peak will be at 10 hertz. But it's kind of cool. One of the things the brain does is it kind of has harmonics, where there'll be amplitudes which are harmonics of this original multiple, which also show little power surges. So 10 hertz is the biggest peak, but you can also see there's a peak around 20, and you can also see there's a little peak around 40. Same here, you can see that the biggest peak is at 20, but if you look, there's also a little peak at 40, which occurs, which is quite common. It's an interesting thing that the brain does, where you can usually see amplitude peaks, which occurs at harmonic multiples of some fundamental or the biggest amplitude peak, and then you'll see little harmonic multiples down the road where there's also littler peaks that occur. And so that is it for the monster event-related potential unit. Here are your 2 in-class discussion questions. We talked a lot about clinical conditions in this chapter. Autism, schizophrenia, dyslexia, or just people with a reading disability. Those are the 3 that I remember off the top of my head. And we talked about how the event-related potential responses between the individuals with these clinical conditions and controls, what those differences are in various event-related potential responses. Just choose one of those examples and describe the ERP measurement that was used. So we have the brainstem response. If your example involves that, then remind me what the brainstem response was or is, and then describe how the 2 groups, your clinical group and the control group, how they differ with respect to the ERP that you're talking about. So that's question 1. It's a little bit more involved. It's kind of a 2-point question. Describe the ERP measurement and then describe how the responses differ between the 2 groups. And then another question, this one's kind of difficult, but it really is getting at one of the key things about this chapter, which is these event-related potential responses. We don't oftentimes know where in the brain they're coming from, because EEG, that is measuring electrical activity from the scalp, does not have very good spatial resolution. We don't really know where these signals are coming from. Nevertheless, researchers use clues which help them make educated guesses regarding where these signals are coming from in the brain. And one of them we talked about is that the P1 event-related potential has sources outside of V1. By the way, those specific regions are oftentimes called extrastriate. Striate means V1. Extrastriate means outside of V1. In the article that you read, ERP Review 1, and I also discussed this article in the previous lecture, there was some evidence presented suggesting that this P1 event-related potential is occurring in extrastriate cortex. But as I said, it's hard to localize EEG signals to specific points of the brain. So my question is what are the clues? What evidence was presented that the P1 event-related potential actually does come from these regions outside of V1, extrastriate cortex? So with that, I am going to turn off the recording. And if anyone wants to stick around and chat, I am more than happy to.